And so our third speaker of the evening is Dr. Kristian Andersen. So he's a postdoctoral researcher at Harvard University and the Broad Institute. And his research looks at how the human immune system impacts the evolution of viruses such as Ebola, and also how the human genome may evolve in response to these infections. So given the devastating impact of the recent Ebola crisis and the lack of approved vaccines or drugs, Kristian's talk is going to cover what we can learn from this epidemic and how we may prevent future outbreaks. So please join me in welcoming Kristian Andersen.
So wanted to talk about Ebola, which is really one of these viruses that lurk in the forest and then, at least until last year, sort of just pops out and infects a bunch of humans and then disappears again. But the way that I started studying Ebola was that I was actually studying another viral hemorrhagic fever. I work at Harvard and the Broad Institute, but we have field sites here in Nigeria and we have a field site in Sierra Leone over here. And we were studying a very different viral hemorrhagic fever called Lassa virus. And Lassa virus is an endemic disease in this part of the world, which means that it's infecting humans every year and very many thousands of people every year. And that was quite unique was that in 2014, or at the end of 2013, we had this Ebola outbreak that came up. And this was the first time that this part of the world had an outbreak of Ebola. And just to show you a little bit about the viruses themselves, as I mentioned that I was studying Lassa virus here, which is an RNA-based virus like Ebola. It only has 4 genes, which is really a very compact virus. Despite that, the case fatality rates for that virus is between 50%. It's 50% in Nigeria, and it's over 80% in Sierra Leone. So it actually kills very many more people than something like Ebola will do, tens of thousands of people every year, yet I bet most of you have never heard about this virus. It's similar to Ebola in the sense that it's a viral hemorrhagic fever. The case fatality rates are the same. The clinical presentations are very similar. But it is pretty different in the sense that Ebola is really a human-to-human transmitted disease. Once you get an outbreak, it's all sustained via human-to-human transmissions, whereas Lassa is more something that comes as a spillover effect every time. And I always like to show this slide when I give my talks because, and I saw one of the sort of sentences in the presentation for today, was that, well, should we in Britain care about diseases that occur outside Britain? And is there any risk of these diseases to actually come to the British shores? And it's a good question, but it's a very naive question because yes, you should. Because the problem is that today, something that occurs in West Africa is not a West African problem, right? We all jet around. I work in West Africa. I just came from New Orleans, now I'm going here. On Friday I go on Milan, then I go to Edinburgh, and I go to Boston, I go to San Diego within 2 weeks. And then I go back to West Africa, and then I get quarantined, and then it's gonna take me a while to get around again. The problem is that these kinds of diseases happening there is not just a problem there, it's a global problem, right? We need to consider these global problems. And when we talk about things like Lassa and Ebola, I think one of the main problems is that they have these extremely stable reservoirs. So for Ebola, we think that they live in bats, and for Lassa, we know that they live in this little cute rodent here. And what is special here is that these reservoirs don't get sick from the disease. So unlike something like bird flu, for example, which kills a large number of birds, these reservoirs don't get sick from the disease, they just keep carrying the virus. And then the main problem is that we don't have any good drug treatments. In fact, we don't have any drugs, and we also don't have any vaccines. So the problem is that the only thing you can do in order to try and stop outbreaks of these is that you have to contain people. You have a case, you put them up in the hospital, you contain them, you go and do contact tracing and make sure if anybody gets sick within the next 3 weeks, you're going to contain those too. And the problem is that you can't just do that because in West Africa they had never seen Ebola before. But let me just show you another problem which is pretty big, which is that this is the GDP of what I call the Mano River Union. The Mano River Union is Liberia, Sierra Leone, and Guinea, and this is where the outbreak happened. The GDP of those 2 countries combined is $11 billion, which might seem like a lot of money, but let's just compare that to the endowment of Harvard, which admittedly is fairly sizable, but it's 3 times bigger, more than 3 times bigger than the combined GDP of these countries. And then the problem is that you have an outbreak, and we basically sit around and do nothing. I'm sure if there had been bombs over there, we would have done something very quickly. But the problem is that the whole worldwide community was just sitting there for more than 6 months doing absolutely nothing about this thing. They can probably deal with it themselves. And the problem is that when you try to do that, well, then something like this is going to happen. Which I just looked at the WHO numbers today, and I believe we have something like 28,000 confirmed cases. The true number of cases of that is going to be much, much higher than that, probably above 50,000 cases by now. And again, with a case fatality rate of about 70%. So it's obviously extremely devastating. And I just wanted to show you some of these photos because they're pretty devastating, but the truth is that this was the reality in West Africa for at least the 6 months where we're sitting back and doing nothing. In fact, this is one of the photos that New York Times took from the hospital that I work with there. And this was true all across West Africa. Where we just let this go on, this was the kind of picture that we had there. So with that in mind, I wanted to talk to you a little bit about the work that we did there. So as I mentioned, we as a team, and it's a pretty large team, but we have worked in Kenema, which is in Sierra Leone, for many years. Since I left Cambridge here about 7 years ago, I have worked together with the hospital in Sierra Leone. And some of you might have heard about Kenema, because Kenema was unfortunately one of these hospitals that got really hard hit by the Ebola outbreak. They were completely overrun with cases. There was one doctor on call who also tragically himself died from Ebola, and he had about 100 patients when he had the most patients in the world. So we have worked there, and when last year we had been following some reports there's this ProMed Mail, and they sort of just describe reports of current outbreaks and weird diseases. And we've been following this for a couple of weeks because it had been mentioning that there was this viral hemorrhagic fever going on in Guinea. And we have worked in that area, and we knew that Lassa fever is hyper-endemic in that region. So we were pretty sure that this was just Lassa fever. But the WHO came out at the end of March, the 27th of March of last year, and they came out and said that actually this is not Lassa, this is Ebola, and this is Ebola Zaire, which is the worst form of Ebola, and also, as I'm saying, is the Ebola that has never been in West Africa. So Stephen and I thought that, well, now we have a field sites there, we have Ebola, we have to get over there immediately. And basically what we did was to say, okay, pack everything we had, took all our trunks, put in personal protective equipment, diagnostic assays for Ebola, and immediately shipped it over there and went over there ourselves. And the main reason for that was this. So this is showing you a map of where the Ebola outbreak occurred. So it started in this region called Guinea, which is on the border of Liberia and Sierra Leone and Guinea. And this is a pretty special region because, there are borders here, but the borders don't mean anything. You have things like a house where they say there's a front doors in Sierra Leone and the back doors in Guinea. So borders don't really apply, and viruses obviously don't carry a passport around. And the main concern that we had to do with Kenema, because Kenema was a referral site for Lassa fever. And Kenema is in fact only one of 2 sites in the world, the other was in Nigeria, one of 2 sites in the world where Lassa fever patients are referred. And that means that Kenema, they get patients from Guinea, they get patients from Liberia, and of course patients from Sierra Leone too. And the main concern that we had here was that, well, but then maybe they're going to get what they believe are Lassa fever patients, but they're actually going to be Ebola patients. They will test them for Lassa and saying, oh, they don't have Lassa, so probably they have malaria, which is, everybody has that in the rainy season. And then that would leave an Ebola case go undetected. And obviously that's not a situation we wanted to be in. So that's why Stephen and I went over there and said, okay, so we need to be absolutely sure that they can definitely do the diagnostics. Because if we can detect it, they can do something about it. And that part of it was successful in the sense that this is Agustin Goba, who is a native Sierra Leonean working in the lab in Kenema Government Hospital. And on the 25th of May of last year, he sent me an email saying, I think we have a case. This is the band. Is this where it's supposed to be if this is a positive assay? And then what I could say to Agustin was that that's absolutely the case. Have you done the controls to make sure that this really is a positive case? And Agustin had done that and went ahead and did a lot of other things. And what this meant was that here, within 12 hours, they had actually detected the first case of Ebola in Sierra Leone, which is important because it's fast detection. And I actually think, so this is the first time that an African country themselves had done the detection. So that part of it, again, was successful and was critical. The problem was that what they then did was they're saying, okay, we have a case in the hospital now, but this case did not come from Kenema. This case was referred from a different hospital. So they sent an outreach team out, went back to the village where that case came from, and this was in Kailaon district in Sierra Leone, which is just on the border of Guinea. And what they realized when they got there was that they actually had a whole cluster of cases. They had 14 cases of Ebola when they came there. And all of these cases could be linked down to this one traditional healer that had been healing people in Guinea and had tragically died from Ebola. And just from that event, you had several further cases. And then what we did with our work, working together with the hospital in Kenema, so all these patients got referred to Kenema, and there was riots and there was all kinds of stuff going on, but the patients were referred to Kenema, the diagnostic was done on site, and then what we do is that we basically get the leftover sample. So the sample that is not being used for diagnostic purposes anymore, we take that sample, we inactivate it, and we send it back to America. And then specifically what we were interested in was that, okay, we're interested in looking at the virus itself. So we're evolutionary biologists, and we look at how do viruses change over time. And then what we was very interested in here was saying, okay, now we have a point introduction of Ebola into a country, and over the next 3 weeks, we got about 70% of all the cases that came into the country. How is the virus changing during the course of those 3 weeks? And now we have since extended that further. We have another about 300 samples or so, which has reached out to January of last year. So, yeah, before I go into that, actually, I just wanted to mention that it is a huge team here, and it's just a couple of the people that are involved in this project. But the first thing that we looked at was saying, okay, so now we have Ebola fever patients coming in on day 1, somebody coming in on day 2, day 3. How is the virus changing during that period of time? Or is it changing? That was actually the first question, because if you looked in the literature for previous outbreaks of Ebola, which have been very short in duration, only a couple of months or so, but if you looked in the literature and people had done this exercise and say, okay, let's look at Ebola on day 1, we sequence it there, and then we look at Ebola on day 60, and we sequence it there, just read the whole genome of the virus, it's identical. And that to us was pretty surprising because it's an RNA-based virus, it's replicating, it's going through humans here, so we would expect this virus to actually mutate. So this was the very first question that we asked. And the answer is that it does mutate, which is exactly what we would expect. And this shows you, just if you look at the black line here, is the number of patients coming in to our study over the first 3 weeks here. And under that is this red line, which is the number of what we call unique viral lineages. And we call something unique, if Oliver just mentioned it has 20,000 base pairs, it's about 18, but 18,000 base pairs, if it's just one mutation anywhere in that genome, then we call this a unique viral lineage. And as we could see was that, okay, the number of unique viral lineages is increasing over time, which means that as we get more patients coming in, the viral population is going to get bigger. And now we are about a year into the outbreak, and we have substantially more data. So now we can actually start to look at, okay, so how does this look, and how do these things connect when we just look at the genomes of these viruses? And this we can get a couple of things coming out of an analysis like this. So one thing is we know that the outbreak started in Guinea over here. So we know that those are the oldest variants of this particular variant of Ebola. And then what happened was that, okay, we have these Guinea strains here. They got into Sierra Leone, and they created this, what we call a cluster 1 of viral lineages. And I should say that the size of the blob here is refers to how many we actually have, how many patients we have with that identical virus. And then every time you see something sprouting out, that's because that one has mutated slightly, usually just by one mutation. So what we could see was that, okay, this lineage 1 then gave rise to lineage 2, and lineage 2 gave rise to lineage 3, and lineage 3 gave rise to lineage 4. So we can now follow, okay, so how is this virus changing over time, and what are these different lineages that we see coming up? Other things you can see is that people that had lineage 3 is very big, right? That blob there is much bigger than any of the other blobs, and that means that you have a lot of patients with that particular lineage of Ebola. Probably what we could determine to be a super spreader event, or something like that. One patient comes in and has infected another 15 patients or so. But what you can also see is that if you look at the viruses that we now find in Liberia and also in Mali, they all come from this lineage 2, which we know occurred in Sierra Leone. So what this tells us is that, okay, the outbreak started in Guinea and actually infected Liberia pretty early on, but then Liberia didn't have any cases. So got from Guinea to Sierra Leone, but then it got from Sierra Leone into the rest of the West Africa. So we think that Nigeria is also lineage 2, so that means that Sierra Leone infected Liberia and Mali, and also all these exported cases, the UK and Germany here, all these cases also come from what we know as Sierra Leonean lineages. And then furthermore, what we can also see is that, okay, there's no reintroduction from Liberia into Sierra Leone, for example, which means that there just seemed to be one event into Sierra Leone, and then it's spreading out from there, but there's nothing coming back into Sierra Leone at that point in time. So, talking about mutations, I don't know about in Europe, but in America, this has been a big question. You alluded to it a little bit, not with Ebola, but with influenza. And I get it often too. And it annoys me a little bit because it's not a very important question. There are questions that are very important, but this, I can guarantee you, is not one of them. And I'll show you in a little bit why. But I just wanted to say that the problem is that when the press has something like, is it going to go airborne, and then everybody in Texas can get scared and go out and buy face masks. But the problem is that that takes away from what the main problem is, right? And the main problem is not whether there's any risk of this coming to America or coming to the UK. The main problem is getting to zero, and getting to zero in West Africa, right? It's in West Africa, we need to get to zero. So that's question number one that's worth discussing. Question number two is that, okay, once, hopefully, we get to zero, how are we going to stay at zero? Right? And I have a lot of science coming in here saying, okay, how is the virus evolving? How are we going to get the diagnostic assays? How are we going to get treatments? How are we going to get vaccines? That will enable us to stay at zero. But the reason why it's a ridiculous question is that this has never happened before. Like, we have many viruses, many viral families, where they have never changed their mode of transmission. Influenza is something, but influenza can already infect via an aerosolized route. It's already airborne. There's some of the viruses that have lost that ability in humans, but presumably because we know they can already do it, it's easy enough for influenza to evolve that. Other viruses never happened. And I just wanted to put into perspective the number of mutations that we actually see. This is the entire tree of Ebola. So the bigger the tree, the more mutations, right? And what you see is that Ebola is smaller than this one. That's pretty easy to see. So this is Lassa over here. Lassa is a huge amount of viral diversity, yet Lassa has never gone airborne. And most viruses actually place over here rather than over here. So even discussing something like Ebola going airborne is just not a very useful question because it's not going to happen. So now we know that, okay, the virus is mutating, the virus is changing, and we can actually use that for something too, because we can look at this diversity and say, okay, let's just try and look back in time and try and address some of the fundamental questions we had about Ebola. And one of the questions we were very interested in was that, okay, so how long has it actually been in West Africa? Did it just, pop up in West Africa, or had it been in West Africa for a long time? And it got particularly interesting because the first paper that was out on this particular Ebola outbreak said that the West African strain, we call this Makona strain, now Makona variant of Ebola, is actually ancestral to all the other Ebola viruses out there. And that means that if that was the case, then Ebola had to been in West Africa for a long time. So that was the first question we did. And this is just to show you some of the data. The virus is very clock-like, which means that we know every year how many mutations would you expect to see in the virus. And we can use that information to actually find out, so when did it split from previous viruses that we have sampled before? And that's what this tree over here is showing, where we're saying, okay, we make a tree where we put all the different Ebola outbreaks on here. And now, because we have a molecular clock, we can go back in time and ask ourselves the question, okay, so this 2014 variant of Ebola, when did it split off from outbreaks, specifically the 2007-2008, which occurred in Central Africa? Right, so now we can say, okay, we know that these two viral variants here split off about a decade ago. And we know that because all previous outbreaks had been in Central Africa, that common ancestor of these two variants would have had to live in Central Africa. So what this tells us is that, okay, this particular variant of Ebola at least couldn't have been in West Africa for more than 10 years. So that tells you something about when, but it doesn't tell you how, because these 10 years would also have to include the virus getting from Central Africa and all the way over to West Africa. And that's several thousands of miles, so it's a very long distance. And the question is, okay, so how did that happen? Did somebody jump on a plane and fly over and then you got Ebola? Probably not. I think the much more likely explanation, that at least that's the explanation I favor, which is that, okay, if we believe that bats serve as the reservoir for Ebola, what you can imagine is that you have an infected bat here that then flies out into the forest, and this bat then meets some other bats and then infects those bats. And those bats then fly back into where they come from, meet some other bats and infect those, and so on and so forth. And what this means is that the virus is spreading via the bats. Right? It's actually spreading through the bats. Right? It's not bats flying from West Africa or from Central Africa to West Africa. It's the virus going through the bat populations that you have across the West African continent. And it does raise a pretty interesting question, because if that's the case, then what about Ebola in all these countries here? Because then you would expect to have Ebola there too. Yet we don't have any human cases of Ebola. But just because we don't see the disease in humans, it does not mean that the virus is not there. And I have one interesting observation, which has to do with the fact that we only observe human cases of Ebola where we also have great apes. Chimps and gorillas only live in the entire world in these two regions. And up until last year, we only had West Africa where we had great apes, but we didn't have any human cases of Ebola. And now we have this, what looks like a pretty perfect overlap between the presence of great apes and also human cases of Ebola. So the kind of thought I'm playing around with here is that, okay, so maybe Ebola needs to get from the bats into the great apes, and we know that great apes are highly susceptible to Ebola, replicates in the great apes, and then maybe jump back into bats or directly into humans. And in this case, at this stage, you actually have an Ebola virus variant which is capable of replicating in humans and capable of causing disease. So with that, I just wanted to finish up with this question, which is that, okay, so now we have had Ebola there. What is the chance of an outbreak happening again? We say, okay, once we get to zero, then we'll be concerned about something else, and then sit back and hope that this is not going to happen again. So the chance of that, I'm going to put at 100%. We know that Ebola is in the environment there now. There is absolutely no chance that we are not going to see further cases of Ebola. It's not going to happen. People are moving further into the rainforested region where we think Ebola lives, and that's not going to stop. So we know that we're going to get cases again. And I think one of the key things that we need to address is that, okay, once we have that first case, well, we need to detect it faster, right? We need to be able to know that it's there, and we need to be able to do something about it. So I just wanted to quote here one of my colleagues, Kristian Andersen, where when we published our paper is that he was saying, okay, our hope is that next time this happens, we will be able to perform deep sequencing right on African soil. And he's talking about deep sequencing here, which is what we did for this study, but really this applies to deep sequencing and also to just diagnostics in general. But I just wanted to show you that deep sequencing is actually pretty interesting and important in this sense because it can be used as a diagnostic. Talk about Derek about finding new pathogens, you can actually use this for pathogen discovery too. And this is why I've spent a lot of my time too. But basically what we get here is that when we have a patient coming in, we take a blood sample from that patient, just take whole blood, take the serum from that, inactivate it, and then we sequence everything that's in there. So what that means is that we take every single nucleotide that's in the blood of that person, and we get these nucleotide sequences coming out of the machine. And what that means is that, okay, if the patient had Ebola, now we can read the Ebola genome. We can also have human reads in there because obviously it came from a human, but we can also see all the other stuff that patient had. So maybe they had E. coli infection, maybe they had malaria infection. We can now see this from this deep sequencing approach. And just to show you some of the data that we did on this was that, well, we sequenced these Ebola patients so we could get Ebola, but we actually also took a subset of 35 patients that were suspected of having Ebola, but turned out to not have Ebola after all. And now we could use this kind of approach to figure out, okay, then what did they have instead? And we saw things like malaria, which was very expected, things like HIV, Lassa virus, enterovirus, things that we know is in the environment there, and that we know actually make people sick. Using these kinds of technologies, we could now get a picture of exactly what did they have. And we have taken that a little further to say we've actually also discovered completely novel viruses. This is just an example of something we published pretty recently where we discovered these two novel rhabdoviruses. Rabies is a rhabdovirus. So rhabdovirus is pretty interesting. And it was only 35% identical to what we already knew about, what was already in the database. So using these types of tools allowed us to find something that we didn't even know was there. And I think it's important because when you come into the doctor today and you tell the doctor that I'm sick, the doctor asks this question: is the patient infected with X, Y, or Z? Is the patient infected with flu, E. coli, Staph aureus, whatever he can think of, sends the blood sample out and tests for those three things. But the problem is that you might not have any of those. Maybe you had something you didn't even test for. And the cool thing about these sequencing-based approaches is that we can ask what is fundamentally a completely different question, and it is, what is the patient infected with? I don't need to test for 3 or 4 or 5 pathogens of interest. I can just test for everything that is actually out there, both known, but also importantly, I think, unknown. So the importance is to get this to Africa, to get these technologies, to get the infrastructure, to make sure that it's there. And just to show you that we are working with partners in West Africa and also in the US, we have what is actually the first rapid diagnostic test for Ebola. So this is a test that is now being rolled out across West Africa, used in clinics, bedside. They can test for Ebola within 15 minutes. But also importantly, we are installing these next generation sequencers in site and in country so they can use those for their clinical purposes. And I just wanted to make that absolutely clear by showing this picture from the clinical lab in Kenema, where we invest a lot of time and money in this. But the problem is, and my concern here is that once we get to 0 cases, most lab will lose interest, pack up the PCR machines and whatever they brought over and go back to Europe, go back to the US and saying, we solved Ebola. And then as I just told you, then we can just sit back, relax, and wait for the next outbreak to happen. And it is going to happen. So that's why it's absolutely critical that we get the infrastructure in place to deal with this. And with that, I just wanted to briefly mention that I had personally, there's 5 of my colleagues Here's Dr. Khan here, who's the head doctor, actually the only viral hemorrhagic fever specialist in Sierra Leone, that all tragically contracted Ebola and died from that. And just the last picture is showing a plaque that you can't see the names here, but there's 47 names on this, which is healthcare workers that lost their lives to Ebola in Kenema Government Hospital alone. So I think for those reasons, even if we're not too concerned about this coming to the UK shores, I do think that this is a question we need to deal with. Thank you.